Here we will see the structure of chromosome. Structure of chromosome seen very visible or very detailed, see very distinct in the phase of metaphase. Metaphase it is the stage of cell division in which chromosomes are arranged at an equatorial plate. Here chromosomes are arranged at equatorial plate called as metaphase. You all know about this metaphase. This metaphasic chromosome is visible, see very distinct. Why it is see very distinct or visible during only metaphase? Because this metaphasic chromosome is highly condensed. It is thick, rod-like, so it is seen under microscope. Here, the structure of chromosome have two similar half, two equal half, two identical structure, this one and this one. These two structures are called as chromatids. These are half of part of chromosome, this one half and this one half, this half structure, half structure. These are called as chromatids. These are identical, similar, so called as sister chromatids. They are called as sister chromatids. Means chromosome have two chromatids called sister chromatids. These chromatids held at a point together. At this point, this called as centromere. Two chromatids held together at a point called as centromere. This structure is called as centromere in both these chromosomes. This centromere have this plate-like structure, this shaped plate, this red color disc shaped plate called as kinetochore. This centromere have disc shaped plate called as kinetochore, to which spindle fibers get attached during cell division. This cell, when there is a cell division, to this centromere, the spindle fiber get attached. Means this is the point kinetochore to which spindle fiber get attached during cell division and get pulled on separate pole, one pole and another pole. Then this is the structure of chromosome, which have a number of parts. One is the centromere, which is also called as primary constriction. Centromere, secondary called primary constriction. This beside this primary constriction, there is a number of other constrictions called as secondary constrictions. Constriction म्हणजे काय? इथे बघा थोडासा क्रोमोझोम काय झालेला आहे, आतमध्ये गेलेला आहे, तो स्वेल नाही आहे, इथे शेप आणि या शेप मध्ये फरक दिसतो आपल्याला, इथे तो कॉन्स्ट्रिक्ट झालेला आहे. This constrictions, first centromere is primary constriction and this is the secondary constriction. This secondary constriction, first some chromosomes have one or two secondary constrictions, few chromosomes only. Here this secondary constriction first, or it is also called as nucleolar organizer. This secondary constriction first called as nucleolar organizer. Why it is called as nucleolar organizer? Because it organizes the nucleolus during interphase. When this metaphase, anaphase, telophase is completed, then there is a interphase occur, nuclear membrane develop, nucleolus develop. This nucleolus is developed or organized due to the secondary constriction. So it is called as nucleolar organizer. This secondary constriction second in some chromosome only, secondary constriction second attached to satellite. Only few chromosomes have secondary constriction second. Here another constriction, but in other chromosome there is no secondary constriction second. Here it is a secondary constriction second to which this satellite body or SAT body is attached. This SAT body or satellite body is small spherical and rounded structure, which is the end of this chromosome, and tip of the chromosome called as telomere. Tip of the chromosome called as telomere. It avoids the sticking of two ends of chromosome together. It avoids sticking two ends of chromosome sticking together. Means it becomes a circular structure, all is the DNA, so it avoids the sticking of two ends. Here these two sister chromatids or this chromosome have the chromatids, and these chromatids have a chromonemata. What is chromonemata? These chromatids have a elongated cylindrical DNA. It is highly coiled. This is cylindrical, highly coiled, long, thread-like. This DNA have this coiling called as chromonema, singular called as chromonema, and number of called as chromonemata. This coiling or folding of this DNA called as chromonemata. This chromatid have double-stranded DNA which is extended from one end to another end of chromosome. It do not have single-stranded, it have double-stranded. Single-stranded only RNA, DNA are double-stranded and they are extended from one end to another end of chromosome. Here, according to position of this centromere, where this centromere is situated, the chromosomes are divided into four types. When centromere present at the center, the centromere is present at the center, both arms are equal, then called as metacentric chromosome. And during anaphase, when chromosomes are pulled to opposite poles, and these chromosomes pulled by spindle fiber, when it pulled, it get this shape. It is attached to spindle fiber, attached to centromere, so it become pulled and two arms are on inner side and it have V shape. During anaphase, this metacentric chromosome become V shape. Here the centromere is present little away from the center, not at the center, it is little away from the center. One arm is small and another arm is long. It is called as submetacentric chromosome. And during anaphase, it become L-shaped. It become L-shaped. Then chromosome is present near the end. One arm is very small and another is long, called as acrocentric chromosome, and during anaphase it become J-shaped. कोणता shape दिसतोय याचा? J-shaped. And last type, when chromosome have centromere and only one arm, single arm, another arm is absent. This centromere is present at the tip, so called as telocentric chromosome, and it becomes I-shaped during anaphase. These are the types of chromosome according to the position of centromere. Here we will see the structure of chromosome and types of chromosome. In structure, there is a two sister chromatids which are identical. They are get attached at one point called as centromere. This centromere have this disc-like plate called as kinetochore, to which the spindle fibers get attached. These two spindle fibers, these two chromatids have constrictions, or this centromere called as primary constriction, and beside this there is a one or two constriction called as secondary constriction. This secondary constriction first called as nucleolar organizer, because it organizes the nucleolus. And in some only few chromosome, this satellite or sat body attached to secondary constriction second. This sat body attached to secondary constriction second only in few chromosome, and the satellite is nothing but the spherical small rounded structure called as sat body, and the end of the chromosome called as telomere. What is the ability of this telomere? It prevents the sticking together two ends of chromosomes. Here there is no smooth structure on the chromosome. There is a number of swellings. These 1, 2, 3. These swellings are called as chromomeres. These swellings are called as chromomeres. And these chromatids have DNA which is extended throughout the length. This DNA is highly coiled, cylindrical, and chromatid have double-stranded DNA which is extended from one end to another end. And according to the position, the chromosomes are of four types. When present at the center, metacentric; from away from the center, submetacentric; near the end, acrocentric; and at the tip, called as telocentric. Here we will see the type of chromosome. Sex chromosomes. In human being, there are 46 chromosomes in each cell. Nucleus have 46 chromosomes, out of which 44 are autosomes and 2 are sex chromosomes. When XX, the female child, when XY, the male child, means XX chromosome form female reproductive system and XY chromosome form male reproductive system. Means these two chromosomes, X and Y chromosomes, responsible for determination of sex of the child. Means it is responsible. It decides sex of the child, so called as sex chromosome, or it is also called as allosomes. These sex chromosomes are of two types. One is X and another is Y. Female having XX and male having XY. These all X are similar, so X and another is Y. Here this X chromosome, it is longer and rod-like, straight. Structure बघायचं कसं दिसतंय, straight, longer and rod-like. Both these are straight, long and rod-like, but type are, this is metacentric, this is acrocentric. X chromosome is metacentric and this Y chromosome is acrocentric. It shows two regions. These are the homologous regions, this below region, and these two regions are called as homologous region. Why called as homologous? Homo means similar. These two segments, these two regions have similar genes, similar location, similar function, so called as homologous regions. And these two regions have dissimilar genes. X chromosome having genes which is not present in the Y chromosome. Y chromosome having genes which is not present in X chromosome. These are the dissimilar genes in this region, so called as non-homologous regions. Here crossing over during meiosis, crossing over takes place only in the homologous regions. क्रॉसिंग ओव्हर जी होत असते मिओसिस मध्ये, झायगोटिक पॅचेटिन आपण पाहिलेले, त्याच्यामध्ये ही जी प्रोसेस आहे क्रॉसिंग ओव्हर ही नॉन सिस्टर क्रोमॅटिड परंतु होमोलॉगस क्रोमोझोम्स मध्ये होते, परंतु इथे नॉन होमोलॉगस मध्ये क्रॉसिंग ओव्हर होणार नाही. In homologous segment or regions where the crossing over takes place. In non-homologous segment, there is a no crossing over takes place. Here, what is the difference between these two chromosomes? Here, the first difference, the X chromosome is straight, rod-like and longer. See here, this straight, rod-like but longer than Y chromosome. Y मध्ये काय लिहाल तुम्ही? It is straight, rod-like but shorter than X chromosome. The another difference, it is metacentric because centromere is present at the center, and this is acrocentric. Y chromosome is acrocentric, this is metacentric. Here, this X chromosome have large amount of euchromatin and small amount of heterochromatin. Euchromatin means extended region and heterochromatin means condensed region. Or euchromatin have large amount of DNA. Large amount of euchromatin means large amount of DNA. So this X chromosome is genetically active. And Y chromosome have small amount of euchromatin, opposite to this small amount. Here metacentric, acrocentric, this shorter, right? And here this heterochromatin is small in here larger, right? This opposite, it is a difference. Here small amount of euchromatin and larger amount of heterochromatin. Means small amount of euchromatin means small DNA. So this is less active or inert. Right? Inactive or inert than that of the less active than, genetically less active than that of the X chromosome because it consists of small amount of DNA. And what is heterochromatin? Euchromatin means portion of DNA. What is heterochromatin? It is a portion of protein where chromosomes get condensed. Means here protein part is larger and DNA part is smaller. But in X chromosome, DNA part is larger and protein part is smaller. So it is genetically active, it is genetically less active. Then these two chromosomes have two regions, homologous region, non-homologous. Why called as homologous? Similar genes, here dissimilar genes. This non-homologous region of X and Y chromosome, here non-homologous region of X chromosome is longer than that of Y chromosome. आणि इकडे काय लिहाल तुम्ही Y मध्ये non-homologous region of Y chromosome is shorter than that of X chromosome. Why it is longer? Because it consists of more genes. Why it is longer? Because it consists of more genes than that of the Y chromosome. This non-homologous region of X chromosome having X-linked genes. And this non-homologous region of Y chromosome have Y-linked genes. Right? These linked genes means they are actually not responsible for sex determination. It is autosomal character, but these genes are attached to sex chromosome and inherit with the sex chromosome, so called as X-linked or Y-linked genes. X-linked genes, color blindness, hemophilia, and Y-linked genes, example Hollandric gene, or it is hypertrichosis. This is the difference and is the structure of X and Y chromosome or allosome.